Okay, so now we're going to have some of the scientists at the Jackson Laboratory and also the New York Stem Cell Foundation talk to us about the details of how this is all going to work. And I have to say that I've been on the board at the Jackson Laboratory for almost 30 years now, and yeah, there's been a lot of change in that time. And I think one of the things that's the most exciting part of this whole thing to me is during this very, very difficult period in life sciences in New York metro area, as we saw when we heard the Newmark report, this is really good news. We have a very large international research organization willing to take a bet on the New York metro area in a big way to make the transformations that Dr. Carden just described. And so, couldn't be a better time. So thank you. All right, I want to introduce Mary Dickinson, who's the Chief Scientific Officer of the Jackson Laboratory, Daniel Paul, who's the Senior Vice President, Discovery and Platform Development of the New York Stem Cell Foundation, and Martin Pera, a professor of the Jackson Laboratory.
Thank you so much, Nancy. It's really exciting to be here. We are thoroughly thrilled about the New York stem cell acquisition, but I will say we are almost equally as thrilled to be in this neighborhood. New York is such a thriving community of research organizations, hospitals, and foundations that really care so much about moving science forward. So we're very pleased to be here, and thank you very much for having us. So we'll start the panel discussion today. This is three scientists talking. So three scientists talking can get pretty hairy pretty quickly. So we're going to start by level setting with what do we mean about stem cells and why are they so exciting? And Martin Pera is one of the world's leaders on stem cell biology, so I'm going to pose a question to him. Can you just give the audience a brief description of what we're talking about when we mean stem cells?
Sure.
And what they're...
So stem cells 101 in 5 minutes. Actually, Lon, you didn't do a bad job. So let's begin with the beginning. What is a stem cell? Well, if you look down a microscope at a stem cell, often it hadn't got many distinguishing features, might just look like a nucleus with a bit of cytoplasm around it. But they've got two key biological properties. The first one is that they can divide and give rise to more stem cells. That's what we call self-renewal. And the second property is that they undergo specialization or differentiation to give rise to the functional cells in a tissue like nerve cells or blood cells, red cells, white cells, platelets. You have stem cells in many of the tissues of your body, and because of those two properties, because they can make more stem cells and give rise to the specialized cells, they're kind of reserve that functions to maintain cell populations that turn over. The lining of your gut, for instance, turns over every 4 days, or in some cases to repair injury or wounds or disease. Now, those are the stem cells in the adult tissues, but generally they have a limited repertoire. In other words, the bone marrow stem cell, the blood stem cell, can form red cells, white cells, and platelets, and that's it. But the cells we're talking today are a very special class of cells called pluripotent stem cells. Now, they were originally derived from very early embryos, first week of development, before any of the specialized tissues of the body have begun to form. And there's just two things you know about them, you need to know. The first is we can grow and expand them indefinitely in the laboratory in that primitive embryonic state. So we can grow them forever. We call them immortalized cells. The second thing about them is all the time they're growing, they remember that property of the embryonic cells from which they derive, the ability to give rise to all the cells of the body. So just as a fertilized egg can form all the cells of the body, so too can pluripotent stem cells. Now, pluripotent stem cells in the human were first described in 1998, and that launched a revolution. Why? Because if you think about those two properties, the ability to grow them, primitive cells in that state forever, and the ability to turn them into any cells in the body means for the first time, we had an indefinitely renewable source of healthy human tissues for use in research or in cell therapy to treat diseases characterized by cell loss or damage that are basically intractable. So as Lon was hinting to you, Nowadays, we can take stem cells and turn them into heart cells and test new drugs on them to see if they have the capacity to cause fatal arrhythmias, rather than doing that experiment in living humans. And we can take these cells, for instance, and turn them into pancreatic islet cells that we can put into patients to treat diabetes. And those trials are going on today. There are over 100 clinical trials now around the world, stem cells in various indications. Now, I'm going to talk about three things that really have propelled that revolution. The first is the discovery that Lon referred to of induced pluripotent stem cells by Shinya Yamanaka, who won the Nobel Prize for that discovery. And as Lon said, what Shinya figured out was you don't really need embryos to make these cells. Through a technique called reprogramming. The technical aspects are simple. I bet I could teach anyone in the room how to do this in a technical sense in a few weeks. You can take a cell from any of you, skin cell, blood cell, and turn it into pluripotent cells with the same properties of the cells that come from embryo. Now, why is that important? Because it opened up a whole realm of human biology that wasn't accessible to us before. So we can make, as my colleagues at New York Stem Cells have done, huge panels of stem cell lines from many patients with disease and use them to study that disease. Or we can make big panels of stem cell lines to do tissue matching, similar to bone marrow banks, so we get around some of the limitations of transplantation that are caused by immune rejection. So that was a huge step forward. The second thing that's really jet-propelled this area is CRISPR gene editing. Because now with CRISPR gene editing, it's very easy to quickly introduce specific mutations into the stem cells. That means we can put in mutations that cause disease, and let's say it's a Parkinson's disease, we can put in a Parkinson's disease mutation, turn the cells into dopaminergic neurons, the cell types is affected, and see how that mutation works. We can use that to study any kind of gene function, human gene function. So that's enormous. The third thing that, again, Lon referred to is organoid technology. I credit the origins of this to Yoshiki Sasai in Japan. There were others. But basically what these people figured out was that if instead of growing the cells in a flat layer in a petri dish, you put them into a three-dimensional system, often in a kind of biological gel, that not only would they make all the cells of the body, but they would shape them, self-assemble them into structures that actually resembled organs, like human brains or human liver. And so they're called organoids, and that added another huge dimension to what we can do because it gives us more sophisticated models that more nearly approach physiology, the things we want to study. So that's kind of where we are, and
Great.
I'll turn it back to Mary.
For those of you with bingo cards out there, there are cells that can be made from any individual, from any genetic background. They can be induced to have mutations that mimic any disease that's genetically induced. They can be differentiated into any cell type in the body, and those cell types can come together to self-assemble into tiny organoids. So 4 really remarkable things about stem cells that make them really exciting for this area. So Daniel, Martin, and Lon both alluded to the special sauce, the power that the New York Stem Cell Foundation has, and Daniel's the architect of the array that you saw in the picture just now. Maybe it would, no, it's gone. In the picture just now, but I want him to tell you in his own words what motivated him to, at the New York Stem Cell, what's been motivating him for the last 14 years and what they really bring to the table that's truly exciting for the relationship that we have now. Daniel?
Thank you. 14 years ago, I did not imagine that we'd be sat on stage as now a part of the Jackson Labs, that's for sure. So I think one of the things that maybe Lon didn't touch on, it was an amazing talk, but maybe the one thing that also maybe needs to be emphasized, though, is if you look around the room, unless you've got your identical twin in the room, none of us are the same. And that genetics that makes us different, whether it's male, female, whether it's where in the world our ancestry is from, also has a huge impact on how diseases develop within the body and how a drug even works within the body. No one single drug will necessarily work in the same way for any one individual, or the side effects of that drug will not necessarily be the same from one person to the next. And so, yes, it's great to be able to screen drugs in cells, but what typically happens, and we do that with different cells in big pharmaceutical companies today, they're not necessarily always using stem cells, but they're using a cell, but it's typically cell lines from 1 or 2 people. And to represent this audience, I can't do that with necessarily 1 or 2 people. And so that's what we were thinking about 12, 14 years ago when APSCs had just come along and we knew that this was going to really change the way we do science. Was, okay, so we have this opportunity. I can take a cell from every single person, a little tube of blood from every single person in this room, and I could make your stem cells. But that's a lot of work. Yes, I could probably similarly teach you in a couple of weeks how to do it, but it's an awful lot of work, and sitting at the bench day in, day out, that takes a toll. And you know, if you're anything like me, and I've had coffee in the morning, and I'm alert and everything, I can focus and I can do everything well. By the end of the day, my attention is drifting, and so maybe I'm making a mistake or I'm doing something slightly wrong. And so we knew that if we wanted to be able to build a technology or use a technology like induced pluripotent stem cells, and be able to do that reproducibly, and do that at the scale that I'm kind of describing, taking a panel like in this room, maybe there was a better way than just throwing sheer brute force human hands at it, because maybe that would cause more problems than we wanted. We've seen other fields be plagued by a lot of manual errors. Things like the cancer field and others have had their issues with where folks have used a cell line they thought was from one person and turned out to be from someone entirely different, and that can have all sorts of confounding impacts. So we made this decision. We said, well, maybe what else is out there that we could use instead of human hands? And so we turned to robotics and automation. And you know, naive folks 14 years ago, we thought it would be as simple as just go to a company, ask them to take some off-the-shelf items, put it together for us, and tell them roughly what we wanted to do, and they would send us some code, and off we'd go. And boy, were we wrong. And it took many, many years really to get it to the state it's in now. And you also don't necessarily from the beginning think about all the other bits that go around something like that. And so needing software development layers where you want to track all the data that's going in and out of a system, or all the data that can then generate downstream. I think we were somewhat naive at the beginning, probably to quite the extent of everything that we would go on to have to do. And so when we started this, we were this small little non-profit foundation. For those of you that don't know us, we'd come out of effectively a kitchen where our founding CEO was impassioned to start this foundation because her son had type 1 diabetes, and she felt that the field really wasn't moving quickly enough in the stem cell space at that time. And so out of that kitchen came this foundation, and from that foundation grew this organization that got us to where we are. And so, and originally we were up at the Columbia Medical Center campus, up in the Alaska building, inside these like small little rooms. We knocked down some walls and we bought all this equipment inside, and we managed to get it working into an amazing facility, even in that location. But we knew to kind of take that to another level, we'd have to go another jump, and we couldn't do it within the space that we were in at the time. And so, thanks to folks that are probably in this room, or folks that many will know in this room, we were able to locate a space at West 54th Street, and working with folks like Taconic and JBB, put together a space that has now transformed a little bit that west side of Manhattan as well. I think we've seen all these biotech buildings pop up, and it's been our home now for the past 8 or so years. And in that space, we've really been able to grow the platform further. And so what this has done for biotech in the city, we've been a little bit of a hub where we've brought together automation engineers, software developers, and biologists, and put all of those folks in the same space to be able to use this platform and the tools that stem cells bring us to be able to hopefully create the data sets that we're going to need to be able to do the things that Lon was talking about and that you've heard about so far. I think I'll add one more bit just at this point, which is, AI is going to be amazing, and it's going to do great things on top of everything that we're doing now. But someone was at a conference, one of the founders of OpenAI, at the end of last year, and one of their slides is stuck with me for a long time, where it said, "Data is the fossil fuel of AI." And that's true, right? Like, you need data to be able to build and train all these models. And what we're missing in the biology space, to be quite honest, is enough data, and enough data that's shared or available to more than just the proprietary group that has generated it. If it stays within that one group, no one's ever going to train a model big enough, because you've got all these other data sets that actually you could tie them together, maybe you could do it. But there's also a problem where some of the data just doesn't exist yet. Like, there isn't simply enough data in the world, probably from diverse, big population scale data sets that isn't just genetics. There's a lot of genetics, but there's a lot of physiological data that we don't have. So building this kind of platform, our vision was always to be able to make large data sets that ultimately maybe we could take advantage of tools like AI.
I think that's such a good point. The data are really the key, and when we talk about that in the synergistic view that Lon presented, having platforms like the platform that's been built at New York Stem Cell Foundation has the potential to crank out gobs and gobs and gobs and gobs and gobs of data. And that's already one level of scalability that's almost mind-blowing. But then when you think about the potential to do in cells, like JAX has done in mice, where we produce the same mouse that can be used all over the world, 10 decades apart, and still get the same results, that accumulation of data now goes beyond mind-blowing. It just blows us off the map. There's just a huge amount of power in that, and we're emerging at a time when AI tools can really take advantage of that level of data power, and we'd really like to see stem cells be used around the globe the way these standard models in mouse have been used to accumulate data and build the knowledge that we need to accelerate discovery. So let's get back to the science and the scientists, and I'll ask Martin this because he deals with these stem cells every day at the bench. What do scientists in the lab really want from stem cells, and what do they want to be able to see and use them for?
Well, so first of all, first and foremost, we want reproducible results because bad data isn't going to help anyone, not even with AI. So that means the tools are very important. We have to have well-characterized, reliable stem cell lines. And what do I mean by well-characterized? I mean, for example, at the genomic level, if we say we've got a disease model with a particular mutation, we have to make sure that cell actually has that mutation and that it hasn't got a lot of other junk going on in its genome that's going to confound our studies. So developing those lines, my colleague Bill Scarns, who's in the audience, has been a leader in taking one particular platform called COLF2 and making a load of neurological disease models mainly from it that are now being used all over the world, and people can rely on them. They can compare the results. Very, very important to have those platforms. But beyond that, we need genetic diversity as well. And the reasons we need genetic diversity are, first of all, scientific, and second of all, ethical. We need genetic diversity because, as has been mentioned, the genetic background of a person has a huge influence on how a disease mutation will play out in their lives. A particular mutation may cause early onset disease in some individuals, but other individuals may not be affected for a long time. So we really need to have that breadth of diversity, and that goes across any kind of functional genomic studies. We really need the diversity. The ethical reason is, if we're going to use these powerful tools to develop new medicines, make new cell therapies, if they all come from one ethnic background, as in white Northern European, then most of the world isn't going to benefit from this stuff because the discoveries won't be necessarily relevant to them. So that's another very powerful reason. And again, my colleagues at Connecticut are now developing very high quality, genetically diverse cell lines. The third thing that people want is patient disease models, and NYSCF has been super active in this. And this is important because if you take something like Alzheimer's disease, there isn't one form of that. The different regions of brains that are affected in individuals can vary, the progression of the disease can vary, the involvement of all the insoluble proteins can vary. So that's why we need large collections from patients, and the good thing about those cells is we know what went on with the patient. We have the record, so we know when we read out something in a cell whether it's relevant or irrelevant to what we're looking at. Now, the last thing is I've been talking up these cell models for all of my career. Now I'm in a position where I have to tell people about their limitations, oddly enough. You've heard all about, well, what do we need mice for anymore? Look, there are some things we can't do with the cells. Diseases involve complex organs with complex functions. So far as we know, there's no brain organoid that exhibits behavioral phenotypes yet, at least so far as I know. Diseases, as Lon mentioned, involve interaction of different physiological systems that are very difficult to capture, even in these multi-organ on-a-chip systems. They're just pick up a textbook of medical physiology, read any chapter, and you'll understand how complex these things really are. The third reason really has to do with, I lost my track of thought. Right, so we're talking about disease models, mice, aging. Aging is very difficult to model in human cells because they don't display the aging phenotype. They're really almost like early fetal cells. So they're not really suitable for studying diseases of aging. So for all these reasons, and last of all, of course, the environment, we can model the acute effects of environmental pollutants in a cultured dish, but long-term chronic exposures or effects of diet or effects of things like exposure to smoke, very, very difficult to model. So we will continue to use mice, but the trick I think came across in Lon's talk is integrating that with a human cell so we get the best possible mouse model of disease.
Yeah, you bring up a really good point. These are cells in a dish, and you think about that, and I know that Daniel has thought about that, and I'm going to turn it over to him in a second. But you think about cells in a dish, and what does a disease mean to a cell in a dish? We know what a disease, looks like in the clinic when it presents to a physician. We know the phenotypes that it would have in the human population, and some of those are recapitulated in the mouse populations. But in a dish, what does that really look like? And Daniel, can you tell disease cells from wild-type cells or non-disease cells in addition? How do you see that evolving as characteristics that we can pick up in culture and utilizing the frameworks that you have and the scale that you have? How do we see bridging that gap and advancing that?
Yeah, it's a good question. So again, there's different levels to this answer. So if I was to have taken some cells from maybe someone that has a heart condition like an arrhythmia, I can visibly see an effect on those cells. So, or maybe like a long QT syndrome where the heart beats irregularly. Because these cells literally are beating in front of your eyes, you look down a microscope and you will see these cells beating. You can then measure the same kinds of things. You can see when it starts to contract too quickly or when it has an irregular sort of beat to it. Those things can be seen, and that's, I'm going to say easy. It's not easy, but it's easier than some things. But a lot of the changes that underpin how a cell looks in a disease state are microscopic, and by just looking at them, we can't see those changes. And so, again, this is where we have used things like AI, because one of the things that I can do on the robots is grow a lot of cells. And so what we've done is take panels, maybe 100 different people, and half those people might be from, say, Parkinson's patients, and half healthy. And I can perturb those cells. I can turn them into neurons. I can then maybe put a drug onto them or just look at them as they are and ask, are there any differences that I can't see by eye? But if I take enough pictures and I have enough replicates and I do this high throughput enough, maybe I can expose this to like a deep learning model or some other advanced analysis system. It doesn't have to be AI, but maybe there are just things that I can measure that I can't see by just looking down a microscope. And when so much of science happens by someone just looking down a microscope, it's easy to miss a lot of things. And so sure enough, we were able to work with Google a few years ago and actually train a model that could make quite accurate predictions about whether, if I show it a new cell, whether that cell came from a healthy person or someone with Parkinson's disease. And actually, in that case, that was just looking at skin cells. So we'd collected thousands of different samples of skin cells. And so we have enough in the biobank to have been able to do a study like that. But that's hard to do. And again, growing some cells, 1 or 2 cell lines, not tremendously difficult, but growing 60 or 100, that becomes a real challenge. And so building these big technological pipelines to be able to do this really is needed. And so one of the things that our mission has always been about is not just doing it for ourselves, though, but has been working with the wider scientific community. So we do an awful lot of collaborations, many places here within the city, Sinai and NYU and Columbia, but really around the world. And so we've shipped, similar to the mice, we've shipped a lot of our cells around the world so that researchers can have access to this. But it's also being able to go across the street, now we can go across the street to Mount Sinai, but go across the city to one of the other institutions and be able to work with researchers. We've opened up this technology platform to allow a lot more than just the researchers within NYSCF to be able to do these things, and we're hoping we can continue to push that forward as well, because this really is going to be how we find new treatments. I think, if you ask me where do I want to be able to, at a point, run a clinical trial in a dish. I think that is, it may sound far-fetched, but it's really not that far away. I can already take 60, 100 different patient cell lines in one experiment and put the same drug onto them and see how they respond differently in a different cell type. The concept of doing this with more cells, or more people in an experiment, or more drugs is not that far away. These are just iterations on what we're already doing. Maybe it's shrinking it to make our experiments smaller and smaller, but the concept of being able to pre-screen a drug in a virtual or in vitro clinical trial before it goes to patients, that's a very real possibility that that's going to happen. In fact, it's going to happen, and it's just a matter of when, not if. And I think this is what's really exciting about where things are heading.
I know many of you in the audience are innovators and are really excited about the innovation landscape here in New York City. And I could tell you in this area, there's no shortage of innovations coming out every day, and an innovative landscape that we can take advantage of to really propel this kind of platform moving forward to the goals that we have. So that concludes the discussion that we had planned. I'm happy to take questions, but I also know that you're running a little bit behind, so I don't want to...
How about one question?
Okay, one question it is. Is there a lucky winner of one question? And it can't be a JAX person. Okay.
Well, if you didn't know that you were living through a historic time that's full of optimism, you should know it now. After the panel discussions that we have had here today. So thank you very much. That is absolutely fascinating.